In this video, we're going to look at transposons. And transposons are elements of the DNA that are mobile. They can move around. And they're common in both prokaryotes and eukaryotes. And one of the things that these transposons can do is that they can influence phenotypes, and they can also influence evolution in the long term. Now, these transposons will code for enzymes that will allow themselves to insert them into their recipient DNA. And to be inserted, the donor and the recipient don't need any homology. However, this transposon event is relatively rare. It will only happen every one in every 10,000 to 10 million cell divisions. So although they are common, they don't happen that often. And so far, we know of 3 different levels of complexity for transposons in prokaryotes. One are the simplest are insertion sequences, or IS elements, and these are the normal constituents of bacterial chromosomes and plasmids. So you're going to find these simple ones in plasmids or the chromosomes of bacteria. Roughly, there are less than 2,000 base pairs. They have your transposable gene, sometimes regulatory genes flanked by short inverted repeats. So here we can see we have beta lactamase, we have a repressor, and then we have our transposase, which will actually do the transposons for us. Now, for these transposons, each side of the transposable element, or the IS element, is going to be flanked by repeated segments of host DNA. And this repeat actually tells us how the transposon was inserted. It suggests that a staggered cut happened that was later filled in. And what I mean by that is, if we look at this image, so here is our DNA that we're going to insert into. So we have a staggered cut to more or less give us sticky ends, if we were talking about the enzymes that we use for PCR. So we're going to create sticky ends here. Then we're going to have an insertion, and here's our inverted repeats. Then we're going to have fill-in and ligation, and you can see that on both sides of our insertion, we have DNA that more or less repeats. So ATT, AT, ATT, AT, TAATA, TAATA. So that's how these group 1 transposons are inserted. Group 2 are more complex, so they will carry genes that are not directly involved in the transposition process. So these are your antibiotic resistant genes. Group 3 are composite transposons, and so you'll have a gene, your central region, that will be flanked by identical or nearly identical IS-linked modules. And they'll have either the same or inverted orientation. And with these transposons, you can transport any sequence in the center region. So it doesn't matter what's in here, you can transport it to some other part of the genome. So let's look at a type of transposon that requires replication to work. So here, let's say we are replicating DNA, and now we have 2 copies of our DNA, purple and green. And we have our transposon here in the purple. So the first thing we need to do is we need to make staggered cuts at the target. So here is our target sequence, and we have made our staggered cut. For the transposon, we make a single cut on each strand. They have to be away from each other on each strand, like shown in this picture. Then each end of our transposon is ligated to a strand of our other version of our plasmid. And what happens is that now we have created replication forks on our purple DNA. So here we can see we have DNA that have open 3' ends, and we have copies of our 5' end right here. So we're ready to copy. So we can now replicate our transposon. So that's what's being shown here in light blue. We have just made a copy of our transposon. And if you follow this image, you can see we took 2 plasmids and made them 1 plasmid. This is called a co-integrate, where you have fused 2 plasmids. However, we don't want 2 plasmids, so we have to separate these eventually. So we need to resolve the co-integrate into 2 separate plasmids. And when we do that, you can see that each plasmid now has its own transposon. And this resolution is coded by the enzyme resolvase. Now, when using a transposon, you can also have inversions, deletions, or rearrangement of the host DNA happen. So let's say you have a transposon and the transposable elements are pointed in different directions. So what can happen is that your different direction transposons can pair up, and then it can be resolved, and your gene, which originally was going from left to right, when this is resolved, it will now switch orientation, where it's now going from right to left. You can also have a deletion. So let's say that your transposons are going in the same direction and they accidentally pair up with each other, and then they are resolved. They will actually cut out the gene that was in between of them. So that's a way that transposons can be used for inversions and deletions. And you can really think of transposons as nature's genetic engineering tools. So antibacterial resistance is usually carried through transposons. You can make new proteins by linking different proteins together through transposable elements. You can have genetic information be transferred through different bacterial species by these transposable elements as well. And when we look at eukaryotic transposons, we can see that they actually look a lot like viruses. So possibly that is where eukaryotes got them from, and prokaryotes as well. It could have been from a retrovirus. And that is it for this video. I will see you in the next video.